Hi everyone, welcome back. An infectious disease has to be transmitted or passed from one host to another in order to make them sick. And the methods of this transmission, what we call modes of transmission, can be categorized into three main groups. You have contact transmission, which includes direct and indirect contact with an infected individual. We also have vehicle transmission, where we can move the pathogen through the air, through the water, through blood or body fluids, through food or other media. And we can also have vectors that will move the pathogens from one host to another, effectively functioning as an intermediate. So this can take the form of a biological vector, in which case an organism has the disease inside their body and will transmit the disease through, for example, biting an uninfected individual. Or they could be a mechanical vector, where a species will literally carry the disease on their body, on their skin or on hair or scales or whatever, and physically introduce it to a new individual. Now, importantly, there is some overlap in some of these classifications. For example, a bloodborne transmission of a disease is also a form of indirect contact with an infected organism. It's their body fluids that have been spilled and then another individual has come into contact with them. So you're going to see overlap in these classifications, but we want to make sure that we can describe them. Describe what contact looks like. Describe what vehicles of transmission look like. Describe what vectors look like and how they work. Now, diseases can be transmitted directly or indirectly using a variety of methods. Direct contact occurs when one uninfected individual makes direct contact with another infected individual, and there is an exchange of some kind. There is contact with infected tissues or fluids. So this can involve skin-on-skin contact, skin-on-fluid contact, sexual contact, an exchange of body fluids. But then we can also encounter the disease indirectly through touching these things without having direct interaction with the infected individual. So for example, if a person blows their nose and has a lot of infectious material in the tissue and throws it on the ground, and then you walk and pick it up and throw it away. Now you have touched a piece of infected material. You have had indirect contact. So touching contaminated materials, contaminated surfaces, eating contaminated food, drinking contaminated water, and so on, all would fall under that indirect category now. An important term to know as we talk about direct and indirect contact is a fomite. A fomite is a material that is likely to carry an infection. This can be clothing or tools or even PPE after we have used it. These materials or structures or surfaces can be a place where these pathogens exist so that other individuals could come into contact with them. Now, even outside of a hospital setting, fomites can cause real trouble. A great example of that would be white nose syndrome in bats. White nose syndrome is a fungal species that infects bats, and it's currently causing a pretty significant epidemic among American bat populations. It's led to significant declines in American bat species, and in some species like the little brown bat, we've seen almost a 90% decline. So the population has shrunk to only a tenth of its previous numbers. White nose syndrome is a disease that mainly spreads through direct contact with infected bats, but it can also spread through fungal spores that are present in the air or on surfaces. And it's particularly concerning that it can spread on surfaces because humans can unintentionally move these spores when we enter caves and move between them. So spelunkers, we think, may have contributed in some way to the spread of this disease by exploring caves and not cleaning gear effectively as they move between caves. In this instance, caving gear can act as a fomite for this disease. Some diseases can be transmitted pretty effectively through aerosols and water droplets. Now, these are little droplets of liquid that are so small they can hang in the air for extended time. And they can be large enough that they can contain bacteria and viruses, and therefore these pathogens can be transmitted through these aerosols and water droplets. These are forms of airborne transmission. The larger the droplet is, the less time it can hang in the air. Larger droplets can hang in the air for a few seconds and then eventually fall to the ground. Smaller droplets can linger for much longer, and these aerosols can remain in the air for minutes to hours in some cases. Many diseases that depend on aerosols to transmit them cause a lot of symptoms that can promote aerosol spray. So for example, diseases that are respiratory in nature, things that cause coughing and sneezing, these can cause more aerosols to be produced, and that can be an effective mode of transmission for them. Essentially, they cause symptoms to perpetuate their own transmission. This is an example of what this can look like. So this is a person sneezing, and you can see all the little particulate that is being sprayed out from their mouth. It can travel quite far. And importantly, we have larger particulate that can move very far but doesn't stay in the air all that long. But then we have the smaller aerosols that are made incredibly fine, and this small cloud can remain hanging in the air for a fairly extended period of time. Infectious disease can vary a lot in its mode of transmission. Some body fluids are much more effective at transmitting some diseases, while others are less effective. And this will vary a lot by the species of the pathogen and its specific adaptations to facilitate that transmission. So for example, body oil, not really that good a way of spreading HIV, but blood and genital fluids are much more effective. This capacity for fluid-borne disease to be transmitted this way is of particular concern for medical staff, people who are going to be working directly with people dealing with their various ailments. There's going to be a lot of body fluid that's produced, and it becomes exceptionally important for that reason to be consistent with PPE and aseptic technique, so that you can minimize the chance that not only that you expose yourself to the diseases that your patients have, but also minimize the chance that you act as a vector, carrying the disease from your patient to other uninfected patients. So vectors are one avenue that we can achieve what we call cross-species transmission, or a zoonotic event. Zoonotic events are really interesting and particularly problematic. A zoonotic event could create a new emerging disease, a pathogen that jumps from one species to another and becomes effective in that new population. Now, these cases could be an example of a new emerging disease where some new mutation, some new strain becomes effective at infecting a new group of organisms, some new collection of species. But it could also be a familiar species of pathogen that can just infect multiple species of hosts and jump between them. Because of the possibility of zoonosis, some human pathogens can also survive in other species. And this can create what we call a reservoir, a source of a pathogen in some other population. In this way, the disease can continue to survive even if we fully eliminate that disease from our population. That disease can still perpetuate because it still exists somewhere in nature. A good example of this is COVID-19. The virus that causes COVID, the SARS-CoV-2 virus, is capable of infecting humans, but it can also infect a variety of other animals, including many cats. So it's possible that one day we figure out how to eliminate COVID completely from our population, but it's still possible for these viruses to exist in populations of other animals, like our house cats, for example. This is particularly problematic because it means even if we get rid of it, we could re-encounter it later. Reservoirs could be living populations of things. It could be wild species or domestic species that exist out there that the pathogen is hiding and circulating in. Or it could be remnants of those species or their waste products or other non-living sources. So with respect to chestnut blight is something that could still exist in the dead stumps of infected chestnut trees. And so even if all of the chestnut trees had died in an area and only their stumps remain, it is possible upon reintroduction of those chestnut trees that they could become reinfected through these persisting stumps where that fungus still exists. Now remember this, zoonoses are any instance where a pathogen jumps species. Zoonoses are possible via close encounters with a variety of species or associate species, organisms that could carry the disease like a vector. or even the waste materials or bedding or burrows or dens of these species. A great example of that would be raccoon roundworm. So raccoons have a parasite that frequently occurs in them called raccoon roundworm. And humans can get exposed to and contract that disease, that parasite, by not encountering the raccoons themselves, but encountering the places where those raccoons are living. So cleaning up a raccoon den, for example, could expose a person to that dangerous parasite, even if they never make contact with a raccoon. A lot of vectors are biting arthropods, especially insects. A lot of these vectors are parasites in their own right. They bite us to steal blood and tissue and things like that from us. They take our resources. And other pathogens piggyback on these species to facilitate their transmission. When a tick bites you, there is the possibility for a fluid exchange. It's not just the tick drinking your blood. It's kind of like they drink your blood and spit a little saliva back up into you. In the process, this small exchange can pass a pathogen from the tick to a person, or from a mosquito to a person, and so on. Now, not all of these vectors are biting creatures, however. There are some non-parasites that can act unwittingly as vectors. Pathogens can persist in other species and then cross over to new species or to familiar old species. These are all examples of zoonoses. And the line between a reservoir of a pathogen and a vector of that pathogen can be blurry sometimes. And there are situations where a pathogen can cause disease both in the host that it infects and the vector that it uses. Now, importantly, once you get exposed to that pathogen, the way that you get exposed, the port of entry that the pathogen uses to enter your body can be very important. These ports or portals of entry are that route of infection. Does it move through a body cavity, for example, the oral or nasal cavities? Does it move through a vulnerable membrane like the eyes or sinuses or your respiratory system? Or does it go through the skin or a breakage in the skin or things like that? All of these are different ports of entry that can be very impactful on the health impacts of that disease. Different pathogens are able to use different ports of entry. Our body varies a lot in its defenses that protect these various ports of entry. So our mucous membranes, for example, are protected by our various mucus secretions, roaming immune cells, and a lot of epithelial layers. Our respiratory tract is protected again by those secretions, different secretions. Our ciliary cells that are actively moving these mucus secretions and removing them from the system. Our cough actions. Our gastrointestinal tract is protected by, again, a lot of mucus, but also the extreme pH of the stomach and even microbes that live naturally and happily inside of your gut and protect it from invaders. So an organism that can bypass these defenses are more successful. That can be a physical or mechanical bypass, like getting injured, getting hurt by a cut or burn, poked by a needle, stepping on a nail, things like that, that can push things past a point of defense. Some species are better able to capitalize on that than others. Or it could be something that the pathogen itself is doing to get around these defenses. Some ports of entry are simply not available to some species, and it really depends on the pathogen's life cycle, their specific needs, their specific adaptations for how they do their infection and how they utilize their host. There's a highly species-specific quality to this. For example, cholera, a bacterial infection, is something that can't be transmitted through nasal droplets. Cholera is a gut infection. It has to exist in the gut or get access to it somehow in order for it to go through its life cycle. Cholera never enters your nasal passage. Something has gone very strange if it has. And not all ports of entry, even the ones that are accessible to different species, are useful to those pathogens. Some ports of entry are dead ends. For example, if you swallowed Staphylococcus aureus, you are probably not going to get a staph infection because staph will die in the acid of your stomach. And other ports of entry are simply a lot more challenging, where this port of entry may still be useful to that pathogen, but because of the biological constraints that the species is under, the process of its infection moves more slowly and it's less effective. The patient may then be at less risk if this is the case, or more able to fight off that pathogen. Or in other cases, the patient risks and symptoms may just be different. They may not be less significant. They may be just as significant and just different. Smallpox is a great example of the importance of ports of entry. Smallpox is a human disease that has been around for millennia. We have mummies that have the scars of smallpox infections that date back 3,000 years. Likely the disease is much, much older. And the disease is really terrible, causes tremendous pain, and it results in death a lot of the time. And if you don't die of the disease, you're plagued by lifelong injury and scarring. However, people figured out that if you exposed a person to smallpox under very specific conditions, you could create a comparatively mild smallpox infection that would then protect them for the rest of their life against that disease. So this process was called variolation. First, you had to extract some fresh pus from a smallpox sore in a live patient. It was really important that the case of smallpox this person had was mild. Then, the practitioner would take that pus and allow it to dry, and then it would be ground up into a fine powder, and then that powder would be administered. It would be either blown into the nose or, much more often, scratched onto the skin. Then, the waiting period would begin, where the patient would wait to develop symptoms. And variolation was this progenitor concept to vaccination. Vaccination was born from the concept of variolation. Now, variolation worked because, first, the dose was very small. It was a very low viral load that a person was being exposed to at any given time. These scabs that a person had when they were dealing with smallpox were places where the immune system was actively fighting. From a mild case, especially, the viral load was low to start, but there is also the presence of a number of antibodies which may have also assisted in this process. Where the powder was administered also really mattered. The disease, we later learned, spreads best through the respiratory system. And so, blowing the powder into a person's nose was usually a pretty bad method of exposure. It was a really dangerous port of entry because the virus really was able to utilize that port of entry very well. Instead, skin contact was more effective because it was less useful for the virus and it produced a slower, more localized infection. It was much more ideal a place to induce infection. The result of a respiratory case of smallpox would be a full body-wide infection. A person would be covered in pox across their entire body. But when scratched onto the skin, that skin infection became localized and it only infected that arm that the person got exposed on.